Hi, my name is Janie Narayan. I am a sophomore chemical and biomolecular engineering major at Johns Hopkins University, and I want to talk about the cancer research that I've conducted in high school as well as currently in college. And I have tried to develop different perspectives when it comes to attacking the cancer issue because I think cancer is a very complex disease. And in order to have any hope at finding a cure or a solution or to characterizing the disease, we have to develop these different perspectives. So the first project I conducted had a more public health perspective and a more chemistry-oriented perspective, and I looked at a biomarker metabolite called nitrosamine in the urine of African American smokers and Caucasian American smokers. And I basically tried to find the difference in the levels of this biomarker in the urine of these two groups of people, and I purified the urine for this biomarker using filtration and purification chromatography, as well as mass spectrometry to actually quantify the levels of this biomarker. And we actually found that esophageal cancer risk is higher in African American smokers. My next few projects were more biologically oriented. So, for example, I looked at a protein called NOXA, which has a domain, a part of the protein that's devoted to a pro-apoptotic function, a pro-cell death function in normal cells, but an anti-apoptotic function in leukemic cells. And I wanted to see the metabolic effects of the other parts of this protein. So I mutated this domain so that it wouldn't work and inserted it into leukemic cells, basically to see the effects of the other parts of that protein. And I did that using three parameters basically. So we were trying to measure oxygen consumption. Oxygen is used in cellular respiration in order to break down glucose into ATP and energy. Lactate production, which is also another metabolic measure, and sensitivity to glucose and glutamine deprivation. So again, glucose is used to break down into energy and ATP, but cancer cells actually divert glucose to a different pathway and instead use glutamine for energy. So basically to see the effects of removing glucose and glutamine on those cells. My current project has a more engineering aspect, so I'm trying to measure the intracellular adhesion forces in between different types of cells, including epithelial cells and cancerous epithelial cells of mammary tissue, of breast tissue, and fibroblasts using imaging technology. And I want to see the effects of mechanical stimuli on the chemical signaling and basically cancer phenotype of these cells. So that's the project I'm currently working on, and I thank you for considering me for the scholarship application.